Hi dear students, a warm good morning to all of you. Welcome to one more session on carbohydrates. In this session, we are going to talk about oligosaccharides and polysaccharides. As we all know, oligosaccharides are those sugars which on breakdown give us 3 to 10 monosaccharide moieties. And based upon that, they can be classified into trisaccharides, tetrasaccharides, pentasaccharides and hexasaccharides when they give 3, 4, 5 and 6 numbers of monosaccharides respectively. And so the first one, the trisaccharide which we have is raffinose, which on splitting gives us glucose, galactose and fructose. And the tetrasaccharide stachyose gives us 2 molecules of galactose, 1 glucose and fructose, whereas verbescose gives 3 galactose, 1 glucose and fructose, and agagose gives 4 galactose, glucose and fructose. These are some of the oligosaccharides. So now let me just talk about the raffinose group of oligosaccharides. The raffinose family of oligosaccharides, they are in fact alpha-1,6 galactosyl extensions of sucrose. As we previously saw, raffinose contains fructose, glucose and galactose. And in this, fructose and glucose together is constituting sucrose. And into that sucrose, where by alpha-1,6 galactosyl extensions, when galactose moiety enters, that becomes members of the raffinose family. So there are mainly two main sugars. There are lots of sugars in the raffinose family, but the most important ones among them are raffinose and stachyose. Now, raffinose is composed of galactose, fructose, and glucose, and stachyose is one more galactose moiety added to raffinose. That means 2 galactose, 1 fructose, and 1 glucose. All these four together constitutes the what? The stachyose sugar. And these oligosaccharides, they are mainly found in molasses as well as in whole grains and all that. You can see sugar bean molasses in some vegetables. Now, for example, in sprouts, cabbage, and all that, you can find these raffinose group of oligosaccharides. So, let me just help you with it. If you're going to take glucose, okay, I'm telling you, this is the whole thing of what? Stachyose. When stachyose undergoes alpha galactosidase breakdown, okay, in stachyose you can see it is a alpha-1,6, it's the one galactose moiety is being attached. In stachyose, there are 2 galactose, 1 glucose and 1 fructose. These whole molecule will constitute stachyose, which contains of 4 monosaccharides. Okay, when stachyose is being broken down by alpha galactosidase, it will result in the formation of raffinose, which is containing 3 monosaccharides. So that is galactose, glucose and fructose. These three together will form the raffinose. Then again, when that undergoes alpha galactosidase, you see, it will result in the formation of sucrose, which contains glucose and fructose. And when sucrose is again degraded or hydrolyzed by the enzyme invertase, you will get glucose and fructose. So the family of raffinose family of oligosaccharides are include stachyose as well as raffinose, as we had mentioned earlier, and they are being formed by the hydrolysis. Now stachyose, now it's in hydrolysis terms, stachyose breaks down by alpha galactosidase, you get raffinose. Raffinose breakdowns by alpha galactosidase, you get sucrose, and sucrose then further breaks down into glucose and fructose. This is a case of, so let's have a look of raffinose, which is consisting of alpha D galactose, alpha D glucose and beta D fructose. The alpha D glucose and beta D fructose is being linked by alpha beta 1,2 linkages, and further alpha D galactose is attached to the sucrose moiety formed here by alpha 1,6 linkages. And the enzyme which is responsible for the hydrolysis also we have already mentioned about this. Then this is coming to stachyose, where 4 sugars are being found to be linked together, where 2 galactose moieties are being linked to 1 sucrose moiety, including glucose and fructose. And stachys, it is a flatulence factor, usually because uh, so it's like a dietary fiber like content, okay, and usually it is not being digested much, so it is used as a flatulence factor. And coming to the significance of these raffinose groups of sugars, this group of oligosaccharides is mainly being found in the plants, and it is known to serve as a desiccation protectant in the seeds. And you can also find that it serves as a transport sugar in the phloem sap, and also as a storage sugar in the case of plants. And they can act as energy metabolites in the plant seeds and as compounds which are being released during the plant germination process. So for the plant to overcome the process of desiccation, stress, as well as when they need to germinate and all that, these raffinose group of sugars play a very important role. And you can find them in beans, cabbage, Brussels sprouts, broccoli, asparagus, and other vegetables and whole grains. And you can find, as I mentioned earlier, it is the alpha galactosidase enzyme which is being responsible for the breakdown of raffinose. But this enzyme is not usually found in the human digestive tract. Now let's move on to the other type of carbohydrate, the polysaccharides. The polysaccharides or the glycans. Now the polysaccharides, they can be divided mainly into two, homopolysaccharides as well as heteropolysaccharides, or they are also called as homoglycans or the heteroglycans. Now if the repeating unit which we consider in the polysaccharide on hydrolysis, if it's the same, then they are being considered to be as homopolysaccharides. But if they are heteromer molecules or different monosaccharide molecules are being formed as a result of using, then you call it as a heteroglycan. Now, polysaccharides, they are polymers with a molecular weight of about 200,000, and they are white and amorphous products. They could be glassy, they are not sweet, they are not reducing, they do not give typical aldose or ketose reactions, and they form colloidal solutions on suspensions. As we all know, starch, cellulose, glycogen, dextrins or inulins, these are all coming under the category of homoglycans, and the heteroglycans do contain mucopolysaccharides. Coming to homopolysaccharides, there are different homopolysaccharides. They are polymers formed by a single type of sugar monomer. Now, fructose, example in the case of inulin, that's an example, or sometimes galactose moieties might be linked together. Now, example as we find in agar, and glucosans in which glucose moieties are being linked together, and you can form starch, glycogen, and cellulose. These are all examples of homopolysaccharides. Now we'll start with the glycogen. Glycogen is the storage polysaccharide which has been found in the animals, and it is mainly stored in the muscle and the liver, and they are present in cells as granules. And it does contain both alpha 1,4 linkages and alpha 1,6 linkages. And so when the glucose moieties are being linked together, they are being linked to each other by alpha 1,4 linkages. Okay, and when a branching comes, the branch point is linked by a alpha 1,6 branch point. And on the hydrolysis, you get sugar, which on reaction with iodine will give a red-violet color. And glycogen can be broken down by both alpha and beta amylases, and also by glycogen phosphorylases. Coming to starch, the next one we have is about starch. Starch is also a storage polysaccharide, but it is mainly been found in the plants. And it consists of two different units. One is, it is consisting of 30% of amylose and 70 to 90% of amylopectin. What is amylose? Amylose is a linear polymer of alpha D glucose, which is being linked by alpha 1,4 glycosidic linkages, and it is soluble in water, reacts with iodine to give a blue color, and the molecular weight of amylose ranges from 50,000 to 200,000. Now, then next we have the amylopectin. Amylopectin is a highly branched polymer which is insoluble in water, and it reacts in iodine to give a reddish violet color. And the molecular weight is between 70,000 to 1 lakh. And at the branches, you can find the alpha 1,6 glycosidic bonds at the branch point. So this is just a previous one, which, as I told you, in starch you have amylose and you have amylose pectin. Alpha glucose units joined by 1,4 glycosidic linkages, and at the branch points we have the amylopectin, which is having alpha 1,6 glycosidic linkages also. So it is in fact the highly branched structure of amylopectin and a helical-like structure of amylose. These are constituents of the starch. This is amylose and this is amylopectin. These two together constitute the composition of starch. And you can see that suspension of amylose and the structure, they will form helical-like conformations. You can have a look over here. Amylose is having a helical-like conformation when they are being soluble in water. And iodine, which will help us to detect the presence of starch, will insert into the middle of the amylose helix to give a blue color, which is a characteristic and diagnostic reaction for starch. Now, the next one I would like to talk about dextrins. What are dextrins? Dextrins are a group of low molecular weight carbohydrates produced by the hydrolysis of starch or glycogen. Let it be, we know that starch and glycogen, they are storage forms of the polysaccharide, but the place where they are like they are being found in plants as well as in animals. So starch is found in plants and glycogen is found in animals. So when that starch is being broken down into smaller parts, okay, low molecular weight compounds of carbohydrates, that they are called as dextrins. So it could be a mixture of polymers of D-glucose units linked by alpha 1,4 linkages or alpha 1,6 glycosidic bonds. Now, how are they being produced? They are mainly being produced by the breakdown using enzymes like amylases. Now, for example, when we are having food within our body, by salivary amylase itself, it undergoes degradation. That is one way the dextrins are being formed. Or in some other ways, like, for example, in the food processing processes like malting and mashing and all that, in that also we will use enzymes so that dextrins are being formed. And the term dextrin itself, of course, different components can come depending upon the level of hydrolysis. Based on that, they are being called as amylodextrins, cyclodextrins, and maltodextrins, etc. Different names have been given to it accordingly. Now, what is the use of dextrin? Dextrin is being used mostly as a glue, and you can use it also as a crispness enhancer for food processing industry, in food batters, in coatings, then in textiles industry as a textile finishing and coating agent to increase the weight and stiffness of the textile fabrics, as a thickening and binding agent in pharmaceuticals and paper coatings, and also dextrin based fibers are also being available as prebiotics. That is also being marketable right now, and it is available in the market. And so you can have white dextrins also and yellow dextrins also. The white dextrins are the ones, as I told you, the dietary fiber ones. The yellow dextrins are also being used mainly in different textile industries and all that. The next polysaccharide which we have is dextran. Dextran is a complex branched glucan, which is a polysaccharide derived from the condensation of glucose, originally derived from wine. Now, according to IUPAC, we define dextran as a branched polyalpha D glucosides of microbial origin having glycosidic bonds, predominantly alpha 1,6 linkages. Now, their length could be of different types. For example, 3 to 2,000 kilodaltons. And the polymer consists of alpha 1,6 glycosidic linkages between the glucose monomers. And the branches are about with alpha 1,3 linkages. We know in the case of dextran, okay, it is alpha 1,6 glycosidic linkages and the branches are alpha 1,3 linkages. But in the case of the other one, now in the case of dextrin, Okay, they are the glucose moieties are being linked together by alpha 1,4 linkages, and the branch linkages are alpha 1,6. So that is the difference between dextran and dextrin. Dextran is of microbial origin, and the linkages which we have between the glucose moieties is alpha 1,6 linkages, and the branches are at alpha 1,3 linkages. Now, dextrin is now produced from sucrose by certain lactic acid bacteria of the family Lactobacillus. Now, you can include Leuconostrep mesenteroides, Streptococcus mutans, and many other varieties of bacteria are also being used for the production of dextran. And coming to dextran's usage, it can be used as plasma expanders in the treatment of shock, used as molecular sieves to separate proteins and other large molecules. They are also being found as components of dental plaques. The next polysaccharide which we want to talk about is cellulose. As we all know, cellulose is a polymer of beta-D-glucose units linked together by beta-1,4 linkages, and it yields glucose upon complete hydrolysis. And the partial hydrolysis of cellulose yields cellobiose, and it is the most abundant polysaccharide which is being found in the case of plants, and it gives no color with iodine. Cellulose is tasteless, odorless, and insoluble in water and most of the organic solvents. This is a structure of cellulose where the beta-D-glucopyranose units have been linked together by beta-1,4 bonds to form the long straight chain strengthened by cross-linking hydrogen bonds. Coming to cellulose digestion, mammals lack the enzyme that hydrolyzes beta-1,4 bonds, so we cannot digest cellulose. It is an important source of bulk in the diet and a major component of the dietary fiber. But the microorganisms in the gut of ruminants and other herbivores can hydrolyze a linkage and ferment the products to short-chain fatty acids as a major source. The next polysaccharide which we have to talk about is about agar. Agar is otherwise known as galactosan, and it is a galactose polymer, and it is obtained from the cell wall of some species of red algae like Gelidium, as well as seaweeds like Sphaerococcus, and which this has a property like it will dissolve in hot water, and when it is cooled, the agar becomes gelatinous. It will be powdery like form, and it melts at a temperature above 60 degrees centigrade, and at a temperature below 40 degrees centigrade, it forms a gelatinous. It will form as a gel, and it's been used in the preparation of culture media for microbiological work, and also been used as a laxative and as a vegetarian gelatin substitute, a thickener for soup, in jellies, ice creams, and Japanese desserts, as a clarifying agent in brewing, and for the sizing fabrics. Another one which we have to discuss is about agarose. Agarose is yet another polysaccharide which has been extracted from certain red seaweed, and it is a linear polymer made up of repeating units of agarobiose, which is a disaccharide made up of D-galactose and 3,6-N-hydrogalactopyranose. And the use of agarose is mainly being used when we have to prepare, now for example, agarose gel electrophoresis for the separation of DNA. This is a DNA separating gel, or you can also use this in the separation of plasmids and all that, DNA plasmids and all that. So with this, we come to end of the session. Thank you.